Example of competitive inhibitor. Inhibition of succinate dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Now, what is succinate dehydrogenase? It is an enzyme. Malonate, it is a chemical that is inhibitor. Now this closely resembles the substrate that is succinate. Okay, so succinate and malonate competes for the active site for enzyme of succinic dehydrogenase. The process is inhibition and the inhibitor is a chemical which is a malonate. So this was the competitive inhibitor. Okay, now how does concentration of substrate affects the enzyme activity and temperature and pH? So enzymes generally function in a narrow range of temperature and pH, what we call the optimum temperature. Now each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Now activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzyme activity, except the thermos aquaticus in biotechnology, because proteins are denatured by heat. Now, concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first, but the reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. Now, this is because the enzyme molecules are fewer than the substrate molecules, and after saturation of this enzyme limited saturation of these molecules, that is enzymes, limited enzymes, there are no free enzyme molecules to bind with the additional substrate molecules. So this is why there is no further rise in concentration of the rise in the velocity of any reaction. See in the diagram.